Hi class, welcome to this first screencast on our unit on genetics. So in this video we're going to cover just the cell cycle and mitosis. In the next video we will cover meiosis, and then in 2 weeks we will cover more in depth into genetics as far as patterns of inheritance. So this is our first sort of time shifting into big idea 3, which is the idea that living systems must have a way to store information, retrieve information, and then obviously transmit that information to the next generation. So we're going to look at how this DNA information is transmitted today. So here's the unit overview for the entire unit. Obviously, genetic information and transmittance of that provides for the continuity of life. So today we'll look at the cell cycle mitosis. How is DNA passed from cell to cell asexually? So hopefully you remember what asexually means. Meiosis is specifically the creation of egg and sperm and how that process leads to greater genetic variation, especially in humans. And then again, in 2 weeks, we'll look at simple and complex patterns of inheritance. So we're really looking at how this heritable information of DNA provides for the continuity of life. Think about how new cells that are constantly being made in your body, how do they receive this heritable information? They have to get that DNA, that code, those instructions from somewhere. And how do they get that? Where do they get it from? Well, the answer is through mitosis and through the cell cycle. So let's just review the types of reproduction. Asexual, this is when the daughter cells, the new cells that are formed, are identical to the parent cell, and there's only one parent cell. Now, unicellular organisms use asexual reproduction to create entirely new organisms. Okay, so this is called binary fission. Binary fission. I'm not going to spell all that out for you. Okay, so for example, a bacterial cell will use asexual reproduction or binary fission to create an entirely new bacterial cell. Multicellular organisms, like plants and animals, are going to use asexual reproduction to grow, to repair damage, to replace body cells. So right now, your body is undergoing asexual reproduction. Kind of crazy, right? Okay, so this is mitosis. Mitosis is creating these new cells through asexual reproduction. Now, the other type of reproduction, sexual, the daughter cells are not identical to the parent cell. There are two parents. And instead of new body cells being formed, what we have being formed are gametes. And gametes are just simply an egg or a sperm. And what happens is when meiosis creates the egg and the sperm, then when fertilization happens, the egg and the sperm come together to produce the zygote, which is the daughter cell. So meiosis creates those gametes. So before we jump into mitosis, let's take a step back and take a look at sort of the life of one of your cells. Every one of your cells has sort of a life cycle, and we call it the cell cycle. This is a complex set of stages. It's highly regulated, and this is another theme, another connection we see in biology. Life has to be highly regulated. So if you can, think about other times in this year already that we've seen that life has to be highly regulated. And so the outcome of the cell cycle is ultimately going to determine what that cell does. Does that cell die, or does that cell go on to create another cell through mitosis? And so here's a figure of the cell cycle in your book. Your Lab 7 packet has an excellent description of it. And of course, your book also has a very good description of it as well. But to begin, your cell starts in G1. So in orange, this olive orange is called interphase. And then in yellow is called mitosis. Now, there are 3 phases of interphase, G1, S, and G2. And G1 is simply when the cell is undergoing normal metabolism. S is when the DNA gets replicated or synthesized. And G2 is when it's preparing for actual division. And then M is obviously division. So let's go through all of these. Interphase, mitotic phase. So when a cell specializes, it's going to often enter a stage where it's no longer dividing. So for example, when a cell becomes specifically a muscle cell, it actually is no longer going through the cell cycle. It's in a phase called G0, but it can re-enter the cycle when given an appropriate signal or cue from the environment. Non-dividing cells may exit the cell cycle, or they might hold at a particular stage of the cycle. And all of this is highly, highly regulated by proteins in the cell. So I want you to answer this question in your notes, and we'll talk about it in class. which type of cell in your body do you think is going to be undergoing the most cell cycles, dividing a lot, and which one do you think is going to be undergoing the least amount of cell cycles? It might be maybe highly specialized or not dividing as often. So going back to the phases of the cell cycle, interphase has 3 subphases. G1 is normal growth and homeostasis and metabolism. S is synthesis or duplication of DNA. And G2 is really preparing that cell to divide, both the cytoplasm and the nucleus. And so the mitotic phase has 2 subdivisions, mitosis, which is specifically the division of just the nucleus, and then cytokinesis, which is the division of the cytoplasm. And they go in this order from G1 all the way down to cytokinesis in that order. Let's talk about how this system, this cycle, is very highly regulated. This regulation system is called the cell cycle control system. It's regulated both internally by proteins and externally by environmental cues. It has specific checkpoints where if it doesn't pass that checkpoint, the cell cycle is going to stop and it's going to maybe go into that non-dividing state, or the cell might die. But if it gets that go-ahead signal, then it's going to go ahead and progress into the next stage. And again, your Lab 7 packet has an excellent description of all of these phases and the checkpoints. So the G1 checkpoint is going to check, okay, are we ready to duplicate our DNA? If not, then we're going to go into the G0 or the non-dividing state. The G1 checkpoint out of all the checkpoints is the most important checkpoint. If a cell goes past that checkpoint, more often than not, it's going to complete the rest of the cell cycle. G2 checkpoint checks, are we ready to divide? Is everything in place? Is our apparatus in place for the nucleus to divide and the cytoplasm to divide? And then the M checkpoint is going to make sure that at metaphase, everything is lined up correctly, and that then we can move on and go into anaphase and separate the sister chromatids. Okay, a little bit more detail about the cell cycle control system. We have proteins called cyclins, and then another protein called cyclin-dependent kinases that control the cell cycle. So cyclins are proteins that control the progression of the cell cycle, and what they do is they bind with these CDKs, these cyclin-dependent kinase enzymes. And you can see that at various points throughout of these 4 phases, different cyclins are at different concentrations. So as cyclin E builds up, that's going to push the cell into S phase to duplicate the DNA. And again, your lab packet has really good descriptions of how cyclins and cyclin-dependent kinases work. What happens when the cell cycle gets out of control and that regulation system somehow breaks down? When that happens, that's uncontrolled cell division, and this leads to cancer. So cancer cells do not respond normally to the body's control mechanism. They don't stop when they're supposed to stop dividing. The cell cycle control system is somehow disrupted. So if some sort of damage happens to a normal cell, as we can see in this figure here, they're going to recognize that damage, they're going to stop dividing, and they're going to apoptose, or basically undergo cell self-destruction. They're going to kill themselves because they know that they're damaged. In a cancer cell, that damaged cell doesn't recognize that it's damaged. It continues to undergo mitosis and divide to create more cancer cells. And we'll talk more about cancer in class. But a little bit more about cancer. They don't need growth factors to grow and divide. Often they might even create their own growth factors. Or they may convey the growth factor signal without the presence of the growth factor even being there. Okay, so growth factors are an important component for normal cells, but not for cancer cells. Okay, so now let's go through the specific phases of mitosis. Remember, there are 2 phases, the division of the nucleus and then division of the cytoplasm. So mitosis passes the complete genome from the parent to the daughter cell. Remember, one parent and the daughter cell is identical. This happens continually in your body to grow, to repair, and to replace old cells. And remember, unicellular or asexual organisms use mitosis to actually create whole new organisms. And the offspring is identical to the parent. And I think you can figure out why that is. And so this is an example of a yeast budding to create a whole new organism. So first, the DNA must be replicated. And remember, when this occurs, during interphase, during the S phase, then the DNA condenses into these discrete chromosomes so that they can eventually line up next to each other and then split into the 2 new cells. And there are 4 distinct phases: prophase, metaphase, anaphase, and telophase. So first we've got to take that DNA double helix, we've got to replicate it during S phase, and then we condense it into these chromosome structures. Now, this chapter and next chapter are very important to understand the difference between these 4 words, and they're kind of confusing because they all start with C. Chromatin is the loose complex of DNA and protein that your DNA is usually in. It's kind of like a ball of yarn, or if you will, spaghetti, like loose cooked spaghetti. It's a mess, it's chaotic, it's not easy to organize. Chromosomes, though, are condensed, they're discrete packets, so maybe they're more like the little macaroni pieces of pasta. They're visible, that's what you can see under a microscope, and they're easy to move around and organize. Sister chromatids are the identical arms of a duplicated chromosome. So once that chromatin duplicates and it condenses, we now see a duplicated chromosome which has 2 sister chromatids. So this is a duplicated chromosome. These arms are called sister chromatids, and they're identical to each other. And then the centromere is right here. This is the region where the chromatids are held together. So these are the 2 pipe cleaners from the lab, and this is the little bead that's holding the pipe cleaners together. So let's go through these phases. So prophase, these 2 round objects here are called centrosomes. And during prophase, the chromatin is beginning to condense into organized chromosomes. During metaphase, the organized condensed chromosomes, now they're called duplicated chromosomes, or they're called sister chromatids. They're going to line up at the metaphase plate, and the centrosomes are now at opposite ends. And this is called the spindle. Okay, this is called the mitotic spindle, and it's made out of microtubules. Next is anaphase. So the sister chromatids are now pulled apart. So this arm, remember, is identical to this arm, but now they're being pulled apart, and they're being pulled to opposite ends of the cell. And it's the kinetochores of the microtubules that are shortening. And lastly, in telophase, we have the decondensing chromosomes now. They're separated from each other. Cytokinesis is already starting to happen, which is the division of the nucleus. And what we see here in an animal cell, this is called a cleavage furrow. And this is only in an animal cell. A plant cell has something different. And so let's take a look at how cytokinesis is different in animal cells. Like I just said, an animal cell has a cleavage furrow, but a plant cell has a cell plate. So what is the difference? Well, the cleavage furrow is pinching in from the outside, whereas a cell plate is actually made in the middle, and it works its way out to the ends. So we have cellulose and cell wall containing material start here, and it actually forms from within. So what happens after cytokinesis? Think about the cell cycle, and think about what the cell is going to then do after cytokinesis, and we'll talk about that in class. Well, actually, I just told you. The daughter cells are then going to go back into interphase, G1, and then S, and then G2. So here is a diagram showing all the stages of mitosis. So after DNA replication, we now have these sister chromatids. They're going to line up at the metaphase plate. They're going to be separated during anaphase. And then now we have 2 cells that are now identical to the parent cell, same DNA. This is an example of a microscope slide of prepared cells. We're going to be doing this in Lab 7 as well. So you should be comfortable with looking at these cells and identifying what stage that they're in. So try to right now identify at least 5 or 6 cells and what stage you think they are in of mitosis. And answer these questions for class discussion.